In this video, we'll go over step 6 of 6.
On the left-hand side, you can go to DNA sequence, import DNA sequences, and you can choose the file from the Teaching the Genome Generation example sequence files from the taster folder. When you open it up, you'll see there's two files inside, KL14F and R. Select both of these and open them up. When we wait a moment, we'll see that both of these have uploaded, and we can open the F sequence first. So this is our forward read, which is what we're going to look at first. And in order to find our first SNP of interest, we're going to search for the reference sequence AGAGGCAG. And zooming into this region, we can see two peaks of about equal strength, a C and a G being called at this locus. In other words, we have an individual that has received a C from one parent and a G from the other at this first SNP. So to find the second and the third SNPs that are within TAS2R38, we need to look at the reverse sequence read because the PCR product for TAS2R38 is longer than the sequence read length. To find the second and third SNPs in TAS2R38, we're going to need to look at the KL14R file, the reverse sequence read that we uploaded. So you can find this in your inventory. Here it is, KL14R. We're going to type in the reverse complement to our reference sequence, which is CACAGGAT. And we can see right here, when we want to actually look at the alignment, we can look at the sequence track. And sometimes when you flip to this window, you have to delete and retype the last sequence there. And instead of looking at the next downstream base, we want to look at the upstream base because we're in the reverse complement. And here we can see an overlapping AG, the reverse complement of which would be TC. So that is our SNP call, is TC for SNP2. When we look for the third SNP, we want to type in GG CTGCA. And this is going to pull us the reference sequence for the third SNP. Flipping over to the sequence track, we can see, zooming in, the upstream base is being called as a CT overlapping, and we can imply that to be an AG heterozygous SNP call. So our example genotype for TAS2R38 is CGTCGA.